Hello, everybody, and welcome to everything you wanted to know about mitosis, but were afraid to ask. Hooray! Look, this smiley face is going to divide into two other smiley faces. Here we go. So mitosis, again, if you recall, is division of the nucleus, okay? And then it's followed by cytokinesis. Okay. During S phase, of course, chromosomes were replicated. We got to get that other set of DNA. So we have a set for each cell. And so when mitosis is initiated, each chromosome has two sister chromatids. So before replication, each chromosome has one chromatid. Then we go through S phase and replicate our DNA. And then each chromosome has two chromatids. Okay, so let's get into mitosis. 5 main phases based on the appearance and the behavior of the chromosome. So you should be able to look at one of the phases and identify it based on what the chromosomes look like. So our 5 phases are prophase, prometaphase, metaphase, anaphase, and telophase. Hopefully you guys have heard these words before. So let's get into it, starting with prophase. The first phase of mitosis is prophase, meaning the stage before. Things that happen during prophase is that basically the chromosomes get ready to sort themselves out. So they go from big, long, loose, stringy piles of spaghetti into those discrete packages that we see as a chromosome. Okay, so spaghetti before, and then into a chromosome. And this is called condensation of chromosomes. So first event, chromosomes condense, and then the spindle microtubules form, and these spindle microtubules will attach to the chromosomes and help guide them to the proper position. And these spindle microtubules are essential, and they are really what makes sure that all the chromosomes get to where they need to be as the nucleus is dividing. Now, we can't get to the nucleus get to the chromosomes because they're in the nucleus, so we have to get rid of the nuclear envelope. So another event of prophase is that the nuclear envelope breaks down, or it starts to break down. And because the chromosomes are condensing, the nucleolus also vanishes. And later I'm going to talk about prophase, prometaphase, and then we'll show some pictures, and then we'll talk about the rest of the phases and show some more pictures. So that's prophase. Prometaphase is the second phase of mitosis. Major events occurring during prometaphase. So the spindle microtubules, again, continue to form and elongate from the centrosome, S-O-M-E, of the cell where the centrioles are located, okay? And they look like a little pair of churros, okay? There are 2 kinds of spindle microtubules that form. One is a polar microtubule, and the other one is a kinetochore microtubule. And the polar microtubules actually do not attach to chromosomes. And the kinetochore microtubules, those are the ones that attach to a chromosome. And they attach to a structure on the chromosome called the kinetochore. We'll see that in a minute, which is a protein-containing structure located at the centromere. So that's a kinetochore. These microtubules grow longer from the poles of the cells from either end of the cell. Think of it as North Pole and South Pole. And they grow towards the nucleus, and they kind of form a basket around it. The nuclear envelope continues to break down so they can reach the chromosome. Okay, and then the chromosomes get captured or are attached to the spindle microtubules as they're reaching down. Okay, just a quick note about centrosomes or centrioles, right? So all cells have a centrosome. Okay, but plant cells and fungi do not have centrioles. Only animal cells have centrioles. But all those cells still form the spindle microtubules. So let's look at some pictures. So this is what the cell looks like in G1. And if you look at the DNA in the nucleus, like I said, it's just a big, huge mess of wiggly spaghetti string. And trying to sort those out when they're all unfolded that way would be very difficult and dangerous to the cell. So here in prophase, those chromosomes begin to condense, to coil up even tighter. The chromatin coils up very, very tight, and you begin to see discrete, recognizable, distinct chromosomes. Okay, and here's our chromosome consisting of 2 sister chromatids. Okay, here we have our centrioles in their centrosome, and here we have the spindle microtubules starting to form. Okay, prometaphase, where's my E? Dropped it down here. So prometaphase, again, the nuclear envelope finishes breaking apart, and the chromosomes have condensed, you see, into these nice little packages, very easy to move around, much easier. We have those polar microtubules here. They're called non-kinetochore. Those are the polar, and they don't attach to a chromosome. And then we have our kinetochore microtubules that do attach to a chromosome. And they attach the chromosome at the kinetochore, which again is a little protein complex that is attached to the centromere. Okay, and in class, I'll show you a little close-up picture of that, so it'll make a little bit more sense. Okay, so good. So now we have all of our chromosomes are attached to kinetochore microtubules, but you see right now they're just kind of all kind of helter-skelter all over the place wherever they condensed. So in order to get ready for metaphase, the microtubules start to position, they start to move the chromosomes around.